to today's video lecture. In today's video lecture, I'll be solving previous year's NEET questions and discussing the answer. Okay, and this is for the chapter Human Health and Diseases, and we are in part 4 of the series. So if you haven't watched part 1 to 3, please do so. Okay, so let's begin. Alright, so question number 1. Interferons are synthesized in response to, this appeared in the year 2001. A. Mycoplasma, B. Bacteria, C. Viruses, and D. Fungi. So interferons are synthesized in response to viruses. Okay, so your answer is C. Please remember that. Okay, so yes, the answer is C. Let's go to the discussion, understand interferons in detail. So interferons are a type of cytokines. Now cytokines are like these chemical messengers that interact with each other. Okay, so between cell to cell there has to be some kind of communication. Okay, so cytokines help in that kind of communication. So for example, interferon is a type of cytokine, okay, and it acts against the cells that are virally infected. So if a virus comes and infects a cell, interferons are released, okay, and they help in destroying the virus, but in turn they also destroy the cell. So that cell has released interferon, and that goes to the next cell, which then activates the processes in the next cell that, okay, there is a viral infection. Okay, so it will help against defending its it helps in defending itself against the virus. Okay, so what happens is that there are receptors present on the cells and or in the cells, and they are called pattern recognition receptors. Okay, this is just for your understanding, not for NEET. Okay, so they're pattern recognition receptors that identify the viral components. Okay, so these receptors are there present on the cells and inside the cells, and they recognize, okay, so this is not our antigen. Okay, this is a foreign antigen that has entered, and this is a viral component. Okay, now the interferons then bind to the membrane of the cells, which induce ribonucleases and destroy the viral and cellular RNA and stop protein synthesis. Okay, so now it has identified that this is a foreign agent, okay, foreign antigen, a viral antigen that has come in, and so the cells begin to synthesize these interferons. Okay, and these interferons then induce the ribonuclease, which destroy the viral RNA but also destroy the cellular RNA, and which in turn then destroys protein synthesis. All right, hence the virus infected cells are prevented from replication and new viral particles are not formed. Now in terms, in also they of course they affect the viral protein formation, so new viral cells are not formed, right? But in also they inhibit normal cellular functions. Okay, so that's actually a disadvantage for the for as humans, for our cells. But of course, if the interferon has to destroy the virus, in turn this is like a disadvantage, that it has to destroy the cell also. So it inhibits normal cellular function so that the virus infected cells are destroyed and the infection cannot spread. So the interferon not just affects the virus, of course that's its job, but it also then begins to affect the normal cellular function. But in turn, obviously the virus will then be destroyed and not move to the next cell and infect fresh new cells. So yeah, this is what interferon does. Okay, so for in terms of NEET, just understand that's a type of cytokine which is antiviral. All right. Okay, so if we've answered the question, I guess interferons synthesized in response to viruses. Yeah, that's the answer. Okay, I guess we can move on then. Question number 2. The antibodies are A. Proteins, B. Carbohydrates, C. Lipids, and D. Germs. And this appeared in the year 1999. So antibodies are proteins. They are glycoproteins. I've already discussed this in the previous lecture, so please watch part 3. Okay, so the answer is A. Antibodies are glycoprotein, that is they contain a protein part, that is 2 heavy chains and 2 light chains. I've also given a diagram of what an antibody looks like with the heavy chain and light chains, and it also has a carbohydrate part, okay, the glycans part. So that is why it's called a glycoprotein. So if they ever ask you if antibody is a glycoprotein, yes it is. All right. So this was simple, and I've already discussed this in the previous lecture, so we'll move on then. Okay, so question number 3. The term active immunity means, and this appeared in the year 1999. A. Increasing rate of heartbeat. B. Increasing quantity of blood. C. Resistance developed after disease. D. Resistance developed before disease. Okay, so the term active immunity. Okay, we already discussed active immunity and passive immunity previously also. So it is a resistance that is developed after the disease. Okay, so your option is C. Alright, so let's go to the discussion. So active immunity is the development of B and T lymphocytes. So basically active immunity is when your immune system is actively working against a pathogen, right? For example, you fall ill, you have a pathogen that's entered your body, and your B and T lymphocytes get activated and they fight against that pathogen to destroy it. Okay, so your immune system has now been activated. Similarly for immunization, like when we take vaccinations, okay, so an attenuated virus is given to us, or a dead virus, a killed virus is given to us, and our B and T lymphocytes begin to activate, and they fight against that, developing antibodies against your memory B and T cells also against it. Okay, so that the cells remember that, okay, so next time now this virus enters our body, we are going to fight it. Okay, so active immunity is basically the cells that are actively working against the pathogen. It's not directly just given to us. It's not passive immunity. Passive is just directly given the antibodies or the antitoxins or whatever to the patient. Okay, but active immunity is when the immune system gets activated and they fight against the pathogen. So when the host gets infected by a pathogen, like I explained, induces the activation of B and T lymphocytes called active immunity, either naturally or via immunization. Okay, immunization when people are vaccinated. Okay, so hence the resistance is developed after the disease. Okay, so this was a good question. It kind of confused you whether it's before or after. So just remember, active means it's the lymphocytes are now working for themselves. Okay, they're not just being given to us. Okay, so this deserves a smiley face. Okay, so but I guess have you answered the question properly? The term active immunity means resistance developed after the disease. Okay, let's move on then. Alright, so question number 4. If a person shows production of interferons in his body, the chances are that he has got an infection of, and this appeared in 1997, A. Tetanus, B. Malaria, C. Typhoid, and D. Measles. Okay, so interferons, like I already discussed, they are antiviral. Okay, they act against viral infections. So tetanus cannot be correct because tetanus is a bacterial disease. B. Malaria, no, it's protozoa, parasite. C. Typhoid, no, that's again a bacterial disease, so it has to be measles. Okay, D. Measles. Answer is D. So interferon cytokines that act against viral infections. So measles is a viral infection, hence the person has production of interferons, okay, to act against the viral infection. Okay, so this was also another good question. It tested you on which disease is viral and which disease is bacterial. Okay, so it was very important to remember that. So yeah, this is important. When I've also discussed previously the different diseases that are there and whether it's bacterial or it's viral. I think part 1 or 2. Okay, we've discussed diseases there in detail. So please remember that. Okay, let's move on. Question number 5. Antibodies are produced by, this appeared in 1996, A. Leukocytes, B. Monocytes, C. Lymphocytes, D. Spleen. Antibodies are produced by lymphocytes. The answer is C. Okay, let's move on. So antibodies are produced by plasma cells. Now how are these plasma cells formed? B. Lymphocytes turn into lymphoblasts and then into plasmablasts, which are precursors for plasma cells. So plasmablasts get converted to plasma cells and then antibodies are formed from these plasma cells. Okay, and then they enter the lymph and the circulatory system. We've already discussed this before. Pretty simple. This is an easy question. Very, very important to remember. If they even write in the options, if it's not lymphocytes, if they write plasma cells, antibodies are produced from plasma cells, you should know yes. If they write B lymphocytes, if they confuse you between T and B lymphocytes, then you should know it's B lymphocytes and not T lymphocytes where antibodies are formed. Okay, so antibodies, according to this question, if you go back, yeah, antibodies are produced by lymphocytes. All right, so let's move on. Question number 6. The interferons are, appeared in 1996. Sorry. A. Antigen properties. B. Antiviral properties. C. Antibiotic proteins. D. All of these. Again, it's an interferon question. So interferon seems like a important topic. It's been previously asked many times. The interferons are A. Antigen properties. No, they are antiviral proteins. Okay, they act against the virus. So the answer is B. Again, I'll not go into details. We have already discussed interferon. You can go back and listen to that part again. Interferons are antiviral proteins. All right, so let's move on. Question number 7. Which one of the following diseases is due to an allergic reaction? Okay, and this appeared in 1995. So we're talking about allergic reaction. So it's A. Enteric fever. No, that's incorrect. B. Skin cancer. No. C. Goiter. No. D. Hay fever. Yes. Hay fever is a type of allergic reaction. So the answer is D. Okay. Let's go to the discussion. So hay fever is an allergen reagent reaction that occurs in the nose. Okay. That's between IgE and the allergen. Okay. The results, now when this reaction happens in the nose, what happens after that? Okay, so there's vascular dilation of the nasal area. Okay, so the blood vessels begin to dilate in that area. Okay, and what happens because of that? It causes increase in capillary pressure and permeability. Okay, so the pressure in the capillary increases and it also becomes permeable. So what happens when it becomes permeable? Fluids begin to leak out. Okay, allowing for fluid leakage in the nasal cavities and deeper tissues of the nose. So the nasal linings also become swollen and secretory. Now antihistamine drugs are administered to prevent the swelling reaction. So whenever there's an allergic reaction, many components are released. Okay, there's histamine from the mast cells and everything. And there are many other chemicals that are released. Many other factors that are released, neutrophilic chemotactic factor and eosinophilic chemotactic factor, and there are various other things. So hay fever usually occurs in the nose after this reaction has happened. Okay, and of course there's permeability of the capillary, the pressure increases, there's increase in permeability, and so then fluids begin to leak out from the capillary, and also the nasal cavity and all area becomes swollen and secretory. Usually antihistamine drugs are given, okay, to control that. Okay, so I've already discussed allergy, I think, in the previous lecture, so you can go watch that. I've discussed that in a little bit of detail. This is only with respect to hay fever. I've already discussed allergen and reagent reaction previously. So if you are lost here, you can watch part 3 and then come back here. Okay, it'll make more sense. Okay, so let's move on then. Question number 8. Antigens are present. This appeared in the year 1995. A. Inside the cytoplasm. B. On nuclear membrane. C. Inside the nucleus. D. On cell surface. So antigens are present. They're not present inside the cytoplasm. On nuclear membrane, no. Inside the nucleus, no. They're present on the cell surface. You should know that. Okay, this is basic. Okay, antigens are present on the cell surface. Like you should know whether that antibodies are glycoproteins. Like similarly, you should know that antigens are present on the cell surface. Alright, so it's an easy question, but you should be aware of that. Answer is D. So a carbohydrate, now let's be more precise, where exactly is it present on the cell surface? So there's a carbohydrate layer, there's a bilipid protein layer there, bilipid layer, and over there is a carbohydrate protein on the outer surface of the plasma membrane. Okay, it forms a coat, and this coat is called glycocalyx. Okay, this coat is called glycocalyx, where the antigen is present. So if they even ask you, if they really want to up the game, and they want to ask you precisely where is antigen present on the cell surface, you should know it's glycocalyx. That's why I'm giving you this information. um, if NEET really wants to mess you up, but yeah, you should know that it's glycocalyx. It also contains, now this layer also contains glycolipids and glycoproteins. Now this layer contains antigens such as MHC, and in erythrocytes they contain the blood group antigen. Okay, so MHC is major histocompatibility complex. Okay, and that are present in cell, that class 1 and class 2. I'm not going to details over here on what MHC is. Maybe I'll discuss it later in much more detail. And erythrocytes, of course, erythrocytes, the ABO blood group system, and so there are antigens present on it. Now where are those antigens present? That's the glycocalyx. Okay, so they contain the blood group antigens. Okay, but yeah, to answer your question, it was fairly simple. You don't need to go into the glycocalyx part and everything. That will come later on when you are studying medicine. Antigens are present on the cell surface. You should know that much. This is basic. Okay, so let's move on then. Okay, so question number 9. A cell-coded protein that is formed in response to infection with most animal viruses is called, this appeared in the year 1994. A, histone, B, antibody, C, interferon, D, antigen. Again an interferon question, so please, I guess, note this down because interferon has been asked multiple times. Okay, so with most animal viruses, yes, of course, it's interferon which act against those animal viruses. Okay, so the answer is C. Okay, again, I will not go into details. So their type of cytokines, I guess, is the same information that I have previously discussed. So I'll not go into so much detail now. Okay, so please go and watch that. Or you can note this down in your notebooks so you remember it when you're studying. Okay. I guess we can move on because we discussed interferon already. Okay, so question number 10. Which of the following is correct regarding AIDS causative agent HIV? This appeared in NEET 2016. Okay, so we come to the topic of HIV and AIDS, which is very important. Which of the following is correct regarding AIDS causative agent HIV? Okay, so to understand HIV, which causes AIDS. A, HIV is enveloped virus containing one molecule of single-stranded RNA and one molecule of reverse transcriptase. No, that's incorrect. B, HIV is enveloped virus that contains two identical molecules of single-stranded RNA and two molecules of reverse transcriptase. Yes, that's correct. B. C, HIV is unenveloped retrovirus. No, that's incorrect. HIV does not escape but attacks the acquired immune response, which is also correct. So there are two answers to this, B and D. Okay, so both are correct, but I guess you can choose one and you probably get the correct answer, the marks for this answer. But yeah, before I directly answer your question, I thought I'll discuss HIV replication in detail. That means the virus itself, how it is structured, and how it replicates in our body. Okay, so it's a very dangerous virus currently. There are many people infected with HIV, and it's an ongoing pandemic which is actually happening in the world right now. And it is obviously there are scientists and researchers in the medical community working actively to find a solution to this to eradicate this virus, but yeah, currently they are still working on it. There are obviously many drugs available, and doctors prescribe those drugs to bring down the effect of AIDS so that a person's life is prolonged. But to completely eradicate or vaccinate yourself against a virus, that has not currently been found. So that's the current situation that we are in. So it's important to understand HIV. Hopefully somebody can find a solution to it. Yeah, but let's get on with this. So now this is an enveloped virus. Okay, you can see this whole envelope here. There's a lipid membrane here, obviously you can see that, and there are glycoproteins here. Okay, so you have GP41, which is this pink part here, and there is GP120, okay, which is the green part here. So this together actually forms a protein GP160, okay. Together GP41 and GP120 forms GP160. So now inside you have this matrix here, okay, you have this matrix over here, you have a capsid here, okay, and you have these two RNA strands here, okay. They are identical to each other, they are just copies, they're not complementary. Please remember that. If they ask you there are two strands which are complementary, no, that's incorrect. They're not complementary, they're just two identical RNA strands, so single stranded RNA strands which are not together, okay. I mean, they're not complementing each other, that's what I mean. And there are two reverse transcriptase enzymes which are attached here, okay, which do the job of replicating, which we will come to later on, okay. So this is the basic structure of the HIV virus, okay. So HIV is an enveloped virus, of course, containing two copies of single-stranded RNA with two molecules of reverse transcriptase. Yes, we've answered that question, as you can see in the structure here. So along with that, now reverse transcriptase is not the only enzyme present there. There are other two enzymes also which play a very important role in the HIV replication, and that is integrase and protease. Okay, remember that. If they even ask you that in the exam, which are the two enzymes are present, it's integrase and protease. Okay, there are two other enzymes present here. Okay, we'll come to the functions later. Now what happens is when HIV enters, okay, it integrates itself into the human genome. Okay, so it is designed in such a way that it forms the single-stranded RNA with the help of reverse transcriptase, gets converted into DNA, and a complementary DNA is formed, and that gets inserted into the human genome. Okay, now once it's entered the human genome, then it is part of the human cell, right? And so then now it will just form viral copies of it. Now it's just using the cell as a factory. That is what the whole point is. That's how the HIV replicates. Okay, we'll come to the details of it, but this is basically how HIV functions. Now HIV, it infects CD4 plus cells. And a CD4, just remember that, I'm not going to detail. CD4 plus cells are receptors. Basically, they're receptors present in certain cells. Now, the HIV virus only infects CD4 plus cells. Okay, so there are certain receptors present called CD4, and they are only present in certain cells, not in all the cells. Okay, so we'll come to what cells they are present in. So CD4 plus cells, I mean, plus is where CD4 is present. Okay, such as monocytes, macrophages, T helper cells, dendritic cells. Now dendritic cells such as Langerhans cells, which is present underneath the epidermis, and follicular cells in lymphoid nodes and spleen. Okay, that's also another dendritic cell. Now also in the CNS, the microglial cells also in the CNS contain CD4. Okay, so these are the list of CD4 plus cells. Now the HIV is only attracted to CD4 plus cells. Okay, it really wants that. It just will go there and infect those cells only. Okay, so it infects CD4 plus cells, and I've given you the list of where the CD4 plus receptor is present. Okay, so even if they ever ask you in the exam what kind of cells, if they give you CD4, you know it's CD4. Okay, but I don't know if they'll go to that extent, because then you will study this later on anyway. Okay, so yeah, so these are the CD4 plus cells that are present in our body, and HIV infects that. Now, how does it infect those CD4 plus cells? We'll come to that. Now GP120, remember that outer layer that I showed you, the protein, GP120 infects the CD4 plus. So GP120 goes and attaches. This is your virus. You have GB41 here, you have your GB120 here, and you have your CD4 plus cell here. Okay, so it will go and attach to that CD4 receptor. Okay, now along with that there is a co-receptor. Now I'm giving a little more detail so that you understand this better and you remember it. Okay, now once GP120 attaches, the head attaches to the CD4 receptor, there are co-receptors also present in that CD4 cells. Okay, now there are different types of co-receptors. I'm just giving you the names, CCR5 and CXCR4. But don't worry about all that. CCR5 is usually present in macrophages, CXCR4 is present in T helper cells, but don't worry about all that. Okay, so once GP120 goes and attaches to CD4 receptor, another arm from GP120 also attaches to the co-receptor. Okay, so this is how it forms the initial attachment. Okay, GP120 attaches itself to the CD4 receptor. There's another arm that attaches to the co-receptor. So it needs two receptors to first bind to the cell. Okay, and that is the initial attachment that the HIV does to the cell. Okay, there's a main receptor, there's a co-receptor. Okay, now once the initial attachment is done, the initial docking is done, GP41 plays its role. Okay, now it's GP41's job. Now what is GP41's job? Is to fuse the lipid bilayer of the virus with the lipid bilayer of the cell. See how the HIV enters into the cell. This is what I'm trying to explain to you. So you have your HIV here, cell here. You have your GP41 here. Make a different color. And you have your GP120 here. Okay, now you have another cell. And remember this, because you will thank me later when you're studying medicine. You will learn the same thing there. Okay, so you have a CD4 receptor here. Okay, this is a CD4. So initial docking happens here. Okay, initial docking happens here. GP120 head attaches to the CD4 receptor. Now I told you there are co-receptors also present there. Okay, it's not just CD4. So let's say CCR5 is here. Okay, CCR5 receptor is here. There's an arm that attaches itself to the CCR5. So this is how the virus acts. Okay, it has an initial receptor which is CD4 and a co-receptor. That job is done. Now it's the job of GP41. Now what does GP41 do? I'll take another color. Now GP41 will fuse this lipid bilayer with this lipid bilayer. Okay, this lipid bilayer is present here and it will fuse with this lipid bilayer over here. It will pull the HIV towards the lipid bilayer of the cell and it will fuse. Okay, and that's how that genome, HIV genome virus enters into the cell. Okay, so this is how it allows for entry into the cell. Once the fusion of the lipid bilayers happens, it creates a gateway. It creates a gateway for the HIV genome to enter into the cell. So this is how the entry is made. Okay. All right. Now once inside, now reverse transcriptase has to do its job. Now understand the central dogma. I hope you know what the central dogma is, how the protein synthesis happens from DNA. Okay, so you have DNA. This is the normal way of doing things. DNA gets converted to mRNA, which is transcription. Okay, and eventually it forms proteins from mRNA, which is, so mRNA contains the code for those proteins. Okay, and it gets translated into proteins. Okay, so this is translation. So it's like trying to solve a clue. Okay, you have a series of codes and you're trying to solve that. Okay, so you have obviously this has already been encoded in us. So the mRNA gets translated into proteins, like a code, like a genetic code is already given to us, and those codes eventually form proteins in us. Okay, so this is how it usually happens. Okay, so now what happens here is we have a RNA with us. Now the HIV is an RNA virus, right? Now RNA has to go back and form DNA. It has to form the complementary DNA of that RNA. Do you get that? So those bases that will be formed will be of a DNA, but complementary to that. Okay, of that particular RNA, it will form DNA virus. So what happens? This is reverse transcriptase. That means it's the opposite way. We don't want to move from DNA to RNA. We want to move from RNA to DNA. Okay. So this is the job of reverse transcriptase. Now, there's one more function that the reverse transcriptase does. First of all, it forms the DNA, single-stranded DNA, but it also destroys the original single-stranded RNA. So initially you have a single-stranded RNA, which is here, and that has formed the complementary DNA over here. It's a single-stranded DNA, single-stranded RNA. But now this reverse transcriptase is there. It begins to destroy the original strand over here. Okay, this is another job of the reverse transcriptase enzyme which is present there. Okay, so there are two functions to this enzyme. Okay, so once that part is done, the DNA is formed. We have a single-stranded DNA. Now the single-stranded DNA converts to complementary double-stranded DNA. It will form the corresponding complementary double-stranded DNA bases, and it will then form the double helical structure which is present in us. So now it is almost like similar to us now. Okay, now it is ready to integrate itself into the human genome. Now the reverse transcriptase has done its job. The virus has entered the cell. The virus has, we understood how the virus enters the cell. Once the virus has entered the cell, it will undergo reverse transcriptase. It will eventually form the double-stranded DNA. Now it is ready to enter the human genome. Okay, so that is where integrase enzyme comes in. Remember I spoke about integrase? That is where integrase enzyme comes in. Integrase enzyme then allows the insertion of the viral DNA into the human DNA. Then integrates itself into the human DNA, which begins to replicate, forming viral components. Now, integrase has done its job. Integrase has allowed the viral DNA to enter into the human genome, human DNA, right? It's now become part of it. Now what's the next step? Once the DNA is formed, it will form mRNA and it will form proteins. Now what is the result of that? There are a whole lot of viral proteins being formed, okay, because of that because of the human viral DNA being part of the human DNA. So it begins to form viral proteins, and which is obviously what we don't want, but the virus has now managed to enter itself, become a part of the human DNA and form viral components of it. Okay, the different proteins that are present in the HIV, and there are genes that are present in the human DNA, in the viral DNA, which has now become part of the human DNA, and they begin to form viral proteins. Now the genes translate into polypeptides, which become functional proteins when protease cuts the chain. Okay, so now obviously the viral components of viral proteins are being formed. There's P24, there's P7, there's P17, there's GP120, there's GP41. P24 is a capsid. Okay, let me go back to the diagram. Yeah, the capsid over here that you see is that's P24. Then there is P7 and there's P17. Okay, these are different proteins. Don't go into all these details. Don't worry about all that. But these are different proteins. Now it has just formed all these different proteins. Now there are genes that are present in the viral DNA. There's GAG gene, there's GAG gene, there's POL gene, there's ENV. I'm just only giving you names, but I'm not going into details of that. This is not necessary, not too much for NEET. I'm just trying to help you understand how the HIV is formed or replicates itself. So now these genes that are present there, they form these proteins. Okay, they form these enzymes, they form the envelope, that's GP41 and GP120. Okay, so now once all these proteins are be formed, that they form as big polypeptide chains. That's the thing. Okay, so now when translation happens, okay, they're big polypeptide chains that are formed. Now these big polypeptide chains are not functional. We can't really do much with those big polypeptide chains. So that's where protease comes in. Protease begins to cut those polypeptide chains, where then after that they become functional. Okay, then after that they become functional proteins, and that's where you can use that. Now along with the functional proteins that are formed, there's the genomic RNA is also formed. We need the HIV genome, right? We have all the proteins with us, which is forming the envelope and all of that. But what about the RNA, the virus? So genomic RNA is also formed from here, which then together form the virus. Okay, so now once the genome has entered into the human genome, the viral genome has entered the human genome, it begins to produce these proteins, these this the genome, the viral genome, over and over again, right? And they combine together to form the virus. Now what happens, that means the cell has now become a factory for producing HIV. That's the whole point. That's where the dangerous part is. Okay, it has now entered the cell and it has used it as a factory to um, produce more and more HIVs. So there is, yes, of course, I already mentioned, genomic RNA is also formed, which combine with all the viral proteins, forming the new mature virion. Okay, so like I said, the genomic RNA along with the viral proteins that are formed together, they all combine. After proteases also cut down all the polypeptide chains to form the small protein chains, they all combine together to form a new HIV virus, which is the mature virus, okay, which then eventually moves out of the cell and begins to attack fresh cells. Okay, it has moved out of the cell and begin to attack fresh cells. So it has used the cell as a factory. So the HIV uses the cell as factories to produce the virus which infect the other cells. Now, destruction of helper T cells reduces the immunity of the patient, leading to AIDS. And the patient eventually is exposed to other infections from bacteria, viruses, and parasites. This ultimately leads to death. Now, this is where the problem is. Now, HIV has entered the genome. It has started replicating, used it as a factory, infecting other cells, and the interesting part is the immune system initially obviously begins to attack the cells. And I'm just telling you how it manages to evade the immune system also. It's just that why we are not able to find a cure, because the GP120 has an antigen cell, a component there, which keeps changing its face. The virus doesn't change, but when an antibody is formed against one particular antigen variant, it will go and attack, but the HIV has the ability to mutate itself. What it will do, it is formed another mutant, it formed another variation or variant. So now the antibody is confused. Okay, this is also ours. Am I supposed to attack this? Antibodies are only specific for particular antigens, right? So antibodies go and affect that particular variant of the HIV, but the HIV has the ability to mutate. That means it can change its face. It can change that antigenic the point there where antibodies attach itself. That is why it is managing to evade the immune system. That's how dangerous HIV is. Okay, so once it enters your system, it is difficult. You can't eradicate it completely from your system. It will remain there. There are drugs obviously present at various points where you can curb the replication, but once it has entered your system. Okay, so this is how bad HIV is, and that's how difficult, that is why it's difficult to form a vaccination against the HIV virus, because there's so many different mutants, there's so many different variants, so many different strains that keep mutating themselves there, that is difficult to form antibodies for a particular variant. Like, how many antibodies are you going to form, right, against that particular variant? So yes, that is one part. But now let's go back to the normal function of HIV, that now it has used the cell as a factory for producing lots of another HIV virus, which then infect fresh cells, and it goes on and on and on, the cycle goes on, right? So now what happens is it also destructs helper T cells. That's where also the problem, that's where AIDS starts. Now AIDS, the full form of AIDS is acquired immunodeficiency syndrome, right? It is obviously infecting all the CD4 plus cells. It is also infecting, obviously CD4 is present in helper T cells, like we already discussed, right? So now once the HIV begins to destroy these helper T cells, it goes in, infects the cells and produces more and more HIV, and then more and more cells are being destroyed, helper T cells are being destroyed. What happens? The immunity is deficient, right? Now helper T cells job is to bring in the cytotoxic cells and help in fighting against the virus, but the helper T cells are not doing the job. Now there's a lot of confusion, there's immunodeficiency, so the immunity of a person becomes weak with time. Okay, as in when HIV begins to spread around the whole body and infect new cells. Okay, so it affects the helper T cells, and helper T cells are being destroyed. It uses them as factories, kills them off, infects another T cell, uses them as factory, kill them off. It goes on. So we're losing T cells. So there's a chaos in the immune system because of that. And obviously the person's deficiency, the defense mechanism goes down. Once your defense mechanism goes down, you can't do anything, you can't fight against the virus. The enemy is going to enter your territory, right, and destroy you. So this is what basically happens. So virus has now entered our territory, and our helper T cells are not able to do their job, and so we are now exposed to other enemies. See, this is what happens. Okay, so once this is very interesting, you can use it as a war zone. It's basically a war zone happening inside you, right? So your helper T cells are destroyed. You've now exposed your territory to other enemies. Now other enemies find it easy to come in, like bacteria and other viruses, and other parasites. They find it easier to come in, so you will basically die of other diseases like bacterial disease or some parasitic diseases. Okay, as HIV is replicating inside you, you've exposed yourself to other diseases, and so you will be immunocompromised. Okay, it's immunocompromised. Okay, so it's an opportunity that other viruses or bacteria, other pathogens or other antigens get an space, a gap to enter into your immune system, into your body, where the immune system can't help you fight against it because it is destroyed. Okay, so this ultimately eventually obviously leads to death because of some other disease. Okay, so it's acquired. Obviously your acquired immune system is being affected. There's deficiency in that immune system. Okay, so this is how HIV and AIDS messes you up. And also now obviously there are drugs, like I previously mentioned, which you will study later in medicine, where it at various points of HIV. Now there's so much research done on HIV and how it is able to evade the immune system. The drugs to stop it at different points. Okay, at different points of the replication. So at least your health will be prolonged, okay, but you can't eradicate it completely from your system. Okay, so yeah, this was an interesting topic, AIDS, but also very very important for exam. Let's come to the exam now for NEET. It's very very important for you to understand this. Yeah. Okay, oops, next question. Let's go back to our question. I think I've explained this. Now once you know this, you can answer any question related to HIV or AIDS, right? So now this should be very easy. Now that you understand, you can come back here and try to answer the question, understand the options that are given below, and now it will be easier for you to understand, right? So let's move on. Okay, so question number 11. HIV that causes AIDS first starts destroying. This appeared in 2015. This is a cancelled exam, and then previously in 2006. Starts destroying the A helper T lymphocytes, B thrombocytes, C B lymphocytes, D leukocytes. So it's helper T lymphocytes. That's A. Answer is A. We've already discussed this. So HIV begins to destroy the helper T lymphocyte, the CD4 plus cells, right? Now you know that. Okay, so this is discussed. We'll move on. See, now it's easier for you to answer these questions, right? Once I've explained HIV replication to you. Okay, so question number 12. At which stage of HIV infection does one usually show symptoms of AIDS? This appeared in 2014. A. Within 15 days of sexual contact with an infected person. B. When the infected retrovirus enters host cells. C. When HIV damages large number of helper T lymphocytes. And D. When the viral DNA is produced by reverse transcriptase. Okay, so symptoms of AIDS are usually seen when HIV damages large number of T helper lymphocytes. So the answer is T. So HIV infection shows AIDS symptoms when a large number of helper T lymphocytes have been destroyed. Like I previously explained, when helper T lymphocytes begin to destroy, the HIV destroys them, your defense mechanism goes down. Okay, you're now exposed to various other diseases, and that's where that is when AIDS is seen. Okay, symptoms of AIDS are seen. All right. Okay, so clear. Let's move on then. Question number 13. Which one of the following statements is correct with respect to AIDS? And this appeared in the year 2010. Sorry. A. The HIV can be transmitted through eating food together with an infected person. No, that's incorrect. So with sharing food, you won't get HIV. Drug addicts are least susceptible to HIV infection, which is incorrect. In fact, sharing needles. So with drug addicts usually tend to share needles to transmit drugs into their system, and if the needle is infected, you got it from an infected person, then you will also get AIDS. Okay, so yeah, that's incorrect. They are more susceptible to HIV infection. C. AIDS patients are being fully cured 100% with proper care and nutrition. No, that's incorrect. The causative HIV retrovirus enters helper T lymphocytes, thus reducing the number. Yes, we already discussed this. So the answer is D. Answer is D. So the HIV attaches to CD4 plus cells, thus using them as factories, thus producing large number of HIV. Okay, we already discussed this. So when they begin to attack T helper lymphocytes, which are also CD4 plus cells, and those helper T cells reduce in number, obviously leads to AIDS. All right, so clear. Let's move on then. Okay, so question number 14. Human immunodeficiency virus HIV has a protein code and a genetic material which is, it's appeared in 1998, a double-stranded RNA, B double-stranded DNA, C single-stranded DNA, D single-stranded RNA. So HIV obviously has a protein code, but the genetic material is a single-stranded RNA. So there are 2 copies of a single-stranded RNA. So the answer is D. Contains 2 copies of single-stranded RNA in its genome. You can go back and look at the structure also when I explain HIV structure and its replication. All right. Okay, this was pretty simple. Okay, question number 15. Which of the following statements is not true for cancer cells in relation to mutations? Submitted in NEET 2016. Okay, so now we are talking about cancer cells and mutations in cancer cells. We'll come to that. We'll explain that there are different genes involved in cancer cells. A. Mutation inactivate the cell control. So to find the statement that is not true, that means it's false. A. Mutations inactivate the cell control. B. Mutations inhibit production of telomerase. C. Mutations in proto-oncogenes accelerate the cell cycle. And D. Mutations destroy telomerase inhibitor. So it has to be B. Mutations don't inhibit the production of telomerase. In fact, it activates the production of telomerase so that there is more rapid cell division happening. Okay, so it is B. And we will come to that. This is a good question with respect to cancer. And I believe when I went through NCERT, it wasn't very explained very well. So that this question would be difficult to answer if you had only read NCERT. So yeah, please note down the points that I'm about to state with respect to cancer cells. Okay, it'll be very important for your exam. So mutations are mutations that occur in the changes that occur in the DNA bases, which are then passed on to the next generation. That means they're passed on to the daughter cells. Okay, when replication of those cells happen. Okay, that's called mutation. That's like permanent. It's always there in you. Okay, so that is so when these mutations occur in some of these genes that are present inside our cells, it leads to formation of cancer cells. Okay, so now there are certain genes called proto-oncogenes. Okay, proto-oncogenes that occur that help in the cell division. So when cell division happens in a normal cell division will happen, but that cell division will only happen for a particular time. Okay, there are a certain number of cells will happen, but there's contact happening between these cells, there's communication happening between these cells. So when they feel that okay, now there's no time, there's no need to divide anymore, it will stop. So there is contact happening between these cells. Now proto-onco genes are present in our cells, which stimulate another gene, which then promotes cell division or cell proliferation, right? Which is a normal functioning of a gene. There is proto-oncogenes allow for cell proliferation to happen. There's a tumor suppressor gene which stops the cell division. Okay, so there are two genes. We are talking about proto-oncogenes. Proto-oncogenes allow the cell division to happen. Now, just imagine if there's a mutation in the proto-oncogenes, what happens? Now, it will not follow its normal function, right? If there's a mutation happen, there's overexpression of that proto-oncogene function. That means it will just keep allowing it to reproduce, okay, divide, I mean to say. So cells are continuously being divided if there's a mutation in the proto-oncogene. Okay, now when the mutation happens, proto-oncogenes become oncogenic genes. Okay, they convert into oncogenic genes. Okay, so obviously this is only one kind of gene which allows for cell division to happen or cell proliferation. So mutation means overexpression of proto-oncogenes. Okay, overexpression of proto-oncogenes is related to continuous cell division. Obviously in normal functioning, what happens is if there is normal cell division happening, there is another gene which stops that cell division, right? There's a tumor suppressor gene which we will study later. Okay, so this results in the proliferation of cells. Other genes such as tumor suppressor gene is also mutated, resulting in continuous proliferation of cells. That means suppression is not happening. That means this under expression of this gene. Okay, there's under expression of this gene. Like proto-oncogenes convert into oncogenic genes and they allow, okay, continuous, just keep dividing continuously, that's fine. But there's a tumor suppressor gene which normally stops the division, but now there's under expression of that because there's a mutation that's happened in the tumor suppressor gene. So it will not stop it. What happens? There's overexpression of proto-oncogenes and there's under expression of tumor suppressor gene. Okay, just remember that. Okay, so this is what's basically happened. This is how a normal cell is converted into cancer cell. It's not just one type of mutation that proto-oncogenes. No, that doesn't work that way. So there are various other mutations that are happening continuously in the cell, which eventually leads to the formation of cancerous cells. Okay, so there's overexpression of proto-oncogenes which convert into oncogenic genes, and there's underexpression of the tumor suppressor gene, which actually normal function is to stop the division. That now we don't need more proliferation of cells. But now if that means if there's under expression of tumor suppressor gene, what happens? The proliferation is continuously happening. There's no stop. That is what leads to formation of tumor, right? So this is another reason, okay, for that to take place. Okay, let's leave it to that. Just won't erase. Okay, so DNA, now I've already spoken of proto-oncogene, there's tumor suppressor gene. Now there is something called a DNA repair gene also which is present. Now every day there is some damage to the DNA happening in our cells, but there is a function to repair that DNA also, right? There's a function to repair that DNA also. Mutation is when the DNA is also not repaired and that is getting carried over, right? So when the DNA repair also is not happening and they are also mutated, okay, which inactivate any mutated DNA repair. Okay, so when DNA repair genes are mutated, what do? They don't repair, they're not able to repair any DNA. So that also gets carried over to the next generation, to the daughter cells, right? There's proto-oncogenes which is mutated. We know tumor suppressor gene is also mutated. Then now DNA repair gene is also mutated. Now DNA repair gene can also not do its job, so it's inactivated, right? So that is another reason, also another characteristic of a cancer cell. Okay, these are all characteristics of cancer cells that I'm talking about. So there's proto-oncogene, there's tumor suppressor gene, there's DNA repair gene which is mutated. All these mutations are happening in our cells. Now there is a function in our cell which is a programmed cell death function. Okay, it's called apoptosis. That means it's like cell suicide. It kills itself. Okay, if it knows that it cannot treat itself, it cannot repair itself, it will kill itself. Now it knows that it is mutated. Then if apoptosis gene is working function normally, what happens? It will kill itself. But okay, there's a lot of mutations, I can't deal with it, I can't allow this to move to the next generation, I'll kill myself. But now there is mutation in that apoptotic gene. Okay, so what happens is the apoptosis genes are mutated to inactivate apoptosis. Okay, so now if that is also inactivated, what happens is it will allow the cell to live. Okay, so anti-apoptotic genes are mutated to allow for survival of cancer cells. Okay, that means the pro-apoptotic genes are mutated to inactivated, that means under expression, there's overexpression of anti-apoptotic genes. Okay, that means they don't allow the cell to even commit suicide. Okay, does not allow it to kill itself basically. Okay, so it's programmed, our cells are programmed in such a way that if there's damage, it kills itself. But in this case, in the formation of cancer cells, these genes are also mutated. So we have proto-onco genes which are mutated, tumor suppressor genes which are mutated, DNA repair genes are mutated, apoptotic genes are also mutated, right? So now in that way it has, captured the whole system. Okay, captured all these genes which do not allow it to kill itself, okay, or even repair itself, and just allow it to continuously proliferate. Now there is a telomerase activity also which is happening inside our cells. Okay, so whenever there's cell division happening, telomerase is present on both ends of the DNA, right? So this is genes are present in this. There's telomerase present on both ends of that gene. Okay, so when there is cell division happening, every time just DNA replication and it goes on, daughter cells are produced and everything, telomerase begins to decrease. This is a normal functioning. Telomerase begins to decrease again. Division happened, again decrease till aging happens of that cell and eventually dies off. Okay, so that is the normal function of the telomerase, telomeres that are present there. Okay, so now telomerase is an enzyme which enhances the telomere. Okay, that means it allows for division to happen up to a certain point till the telomere ceases to exist or becomes shortens in length and eventually dies, which is called aging of the cell, senescence. Okay, but now in cancer cells, it enhances the telomerase activity. That means keep the telomerase activity there, keep the telomeres there in the chromosome to allow for continuous replication. Okay, this is the function of telomeres, telomerase and telomeres. Okay, so if telomerase activity is enhanced, what happens? The telomeres will still be there. There will not be decreasing of length of telomeres on either sides of the genes, and there will be continuous proliferation, continuous division. So the cell is now confused. It thinks the telomeres are still there. We need to still keep producing, right? But obviously in a normal function, the telomeres reduce in length and eventually it stops division. Okay, which is a normal function. But in cancer cells, telomerase activity is enhanced. Okay, which allows for continuous proliferation of cells. All right. Now, cancer cells also have the capacity of forming, now this is another characteristic of cancer cells, they have the capacity of forming new blood vessels. Okay, via the help of angiogenic factors. Okay, just to let you know, now if tumor cells are being formed here, it has the capacity of forming new blood vessels to supply blood. Now it needs to survive. Okay, now it's become like a tumor. It's a cancer cell that's present in one place in our body. Okay, all these mutations have happened, like I already previously spoke to you about. Now it also has the ability to form new blood vessels to allow for blood for nutrition. Okay, well, obviously blood has a lot of nutrition, right? So it takes the nutrition from us, okay, to help it live. So that's another characteristic of cancer cells. Now cancer cells also have the capacity to invade and travel to the other regions of the body, which results in malignant tumor. Okay, cancer cells have the capacity to invade That means once the tumor has been formed, the cancer cells have been formed, they invade and they have the ability to move to other parts of the body. Now, when that happens, that's a dangerous situation, because now the cancer cells are spreading all over your body from one position. Like you have cancer of the skin, okay? Now it is lodged here, it's there inside, it's benign, it's fine, we can still treat it. But once it slows off and begins to invade and into other parts of the body and settles there and begins to proliferate over there, then that's called metastasis. Okay, that's called metastasis and results in malignant tumor. Okay, now cancer cells also have the ability to evade attack from immune cells. Okay, obviously immune cells will think that something is wrong, this doesn't seem right. Okay, these are not our because they're all mutated genes, so the protein that will be formed will be different proteins, not our proteins that don't belong to us. So they also have the capacity, they are designed in such a way that they also evade the immune cells. Okay, so this is another dangerous thing. That is why a lot of treatment is done by surgery, by radiotherapy. They can remove cancer cells, tumor cells from it, because our immune cells are not able to actively function against these cancerous cells. Okay, so cancer cells also have the ability to evade attack from immune cells. Sorry. So these are the characteristics of cancer cells, and I would suggest you note this down, please, because this is very important. This was actually a good question, because it will be very difficult for you to answer this. None of it is mentioned in NCRT clearly. They just spoke about proto-oncogenes, and which is not really the only reason why cancer cells are formed. But there are various other mutations that are happening inside. So cancer can also be due to viral infection. It can be due to exposed to radiation through some carcinogens, chemicals that are carcinogenic. They can form cancer cells. So there are various substances that are carcinogenic, and you use them on a daily basis, and it could lead to cancer. It could lead to mutations inside your genes. So this is very important to know. Without this, you wouldn't be able to answer the question. So this was a very good question that they've asked. And yeah, it's not true for cancer cells. Yeah, it's inhibit production of telomerase. Now you know why, right? So yeah, let's move on then. Okay, so question number 16. Which one of the following is not a property of cancerous cells, whereas the remaining three are? This appeared in the year 2012. They compete with normal cells for vital nutrients. They do not remain confined in the area of formation. C, they divide in an uncontrolled manner. D, they show contact inhibition. Which is not a property is obviously D. Sorry. Obviously D. A, B, C are all properties of cancerous cells. Answer is D. So cancer cells don't show contact inhibition unlike normal cells. So when normal cells divide, they communicate with each other, right? So there is a certain limit to how much proliferation will happen. The cancer cells don't do that, because obviously mutation has happened, like I previously explained, right? All these mutations have happened in the genes. Cancer cells just continuously begin to divide, and that contact inhibition is gone. Alright, so that's why they don't show contact inhibition unlike normal cells. Okay, so they have the ability to continuously divide. Okay, so question number 17. Which one of the following techniques is safest for the detection of cancers? This appeared in NEET 2010. A, MRI, B, radiography, X-ray, C, CT, and D, histopathological studies. Okay, so the safest technique is MRI, magnetic resonance imaging. Answer A. So MRI is the safest technique, and it's used to detect cancer of internal organs. So it uses magnetic fields and non-ionizing radiation to detect pathological and physiological changes. Okay, so MRI is a useful technique, and it's the safest technique for detecting cancer, especially of the internal organs. Okay, next question. Question number 18. Carcinoma refers to, and this appeared in 2003, A. Malignant tumors of the connective tissue. B. Malignant tumors of the skin or mucous membrane. C. Malignant tumors of the colon. And D. Benign tumors of the connective tissue. So, carcinoma refers to malignant tumors of the skin or mucous membrane. Okay, answer is B. So, carcinoma is malignant in nature. Okay, so answer is B. Okay, it arises from an epithelium. Like I said, it's a malignant tumor and it arises from an epithelium. So, if the carcinoma is related to the epithelium. If it arises from squamous epithelium, that is squamous cell epithelium. Sorry. This would be carcinoma. Okay, and if it arises from glandular epithelium, it is called adenoma. Okay, so if it's squamous epithelium, it is squamous cell carcinoma. It's glandular epithelium, it is called adenoma. Okay, so different terms given to this. Okay, if it is sarcoma, if they ever ask you what is sarcoma, what type of tumor is that, or what type of cancer is that? So it's a malignant tumor of the connective tissue. Okay, of connective tissue. All right. Carcinoma is a tumor that malignant tumor that arises from an epithelium. Okay, let's move on. Okay, question number 19. Cancerous cells can easily be destroyed by radiations due to it's appeared in the year 2002. A. Rapid cell division. B. Lack of nutrition. C. Fast mutation. And D. Lack of oxygen. So cancerous cells have the ability to continuously divide, right? So they are destroyed by radiation due to rapid cell division. Answer is A. Although they could have termed this phrase this question a little differently, but whatever, you have to deal with what we have, right? So cancer cells continuously divide, okay, and when they undergo the cell cycle division, the radiation therapy affects its ability to divide by damaging the DNA and hence kills it. So the amount and like I said, the amount and type of radiation and the speed of cell growth determines whether the radiation will be able to kill all the cells. Okay, so so because of its rapid cell division ability, the amount of radiation and its ability to grow or to divide rapidly, that it will determine whether the cells will be killed or not. And obviously radiation therapy is used to damage the DNA of the cell so that it stops dividing. Okay, let's move on then. Question number 20. Reason of lung cancer is, and this appeared in 2001, A. Coal mining, B. Calcium fluoride, C. Cement factory, D. Bauxite mining. So reason of lung cancer is A. Coal mining. Okay, answer is A. So obviously the miners that work in the coal mining industry, they're continuously exposed to this coal dust, which is a carcinogen. So once you keep inhaling that dust over and over and over again, it leads to mutation in the cells, which eventually leads to cancer, lung cancer. Okay, and so it's very important to take precautions. The miners that are working in the industry and those that are associated with the industry should make sure that the workers are not exposed and they're protected. Okay, so that's a long shot. But coal mining, it has been found that coal mining workers do experience lung cancer due to prolonged exposure to coal dust. All right. Okay, question number 21. Which of the following will be curable in next 2 decades? Okay, this appeared in 1997. So you have A. Tuberculosis, B. Cancer, C. Poliomyelitis, and D. None of these. Okay, so this is a very general question that they've asked, but it's a self-aware question. But you know that tuberculosis and polio vaccines are available, which can prevent you from tuberculosis and polio. Cancer, currently, we don't have any vaccine against cancer or any short treatment that will treat cancer completely, because many people die because of cancer. But there is obviously, on the bright side, there is continuous research happening, and from the scientific community, the medical community, it provides its data, to the researchers, so that there are proper good treatments available to cure cancer. So if in the next 2 decades, which is within 20 years, we can hope that yes, there will be a curable, where the cancer will be curable, there'll be a proper medicine available to resolve this problem. Okay, so you can say B is the right answer for this. Answer B. So yeah, with progress in cancer research, there's hope that there will be cure to cancer. Okay, which is a very vague and a very general question that they've asked. But anyway, if you choose an answer, then yeah, maybe cancer medicine would be available in the next 2 decades. Okay, so we've come to the end of this video lecture, and I hope you've enjoyed today's video lecture. I hope you've learned something new today. And don't forget to like, share, and subscribe to MG NEET Biology. And I'm available on Facebook, Twitter, and Instagram, so you can follow me there for more updates. And I'll see you in the next video lecture. Right? So bye.